Good day everyone, my name is Rizka and my peers are Ferren, Wiranata and Anil, and today we will be presenting of the paper that we picked, which is Discovery and Characterization of Tubercidin Biosynthetic Pathway from Streptomyces tubercidicus NBRC 13090. And this is the outline. First off, tubercidin has a procremidin or 7-diazepurin, and this compound is known as a secondary metabolite. This family of secondary metabolites is known for its diverse biological functions. Tubercidin itself is known as an analog. It is a powerful RNA and DNA polymerase inhibitor and has antimicrobacterial, antiviral, and antitumor activity. Using an unusual diazepurin core, according to the paper, which over here is supposed to contain an N, but instead it is a C. And what the paper wants to know is the molecular logic behind the formation of the compound itself, in hopes that for future studies it can help in terms of finding new antibiotics using tubercidin. These are methods. First is sequence analysis of the genome as tubercidicus, and then from that sequence analysis they put it into the gen bank, which will get them the session numbers, so that they form a genomic library construction and screen for Streptomyces tubercidicus, and then to make sure they can produce tubercidin, they ferment the Streptomyces strains, and then they overexpress and purify tub-E, tub-D, and tub-G, which are proteins, in E. coli ricida via genetic engineering, and then they conduct an enzymatic assay of tub-E, tub-D, and tub-G, and finally liquid chromatography and mass spectrometry analysis.
So moving on to the results, the discovery of the gene cluster for tubercidin biosynthesis goes like this. Tubercidin possesses a diazepurin chlore, which is similar to toyocamycin and sengimavamycin. So the logic goes that the enzymes in the tubercidin production must be similar to these two antibiotics. Thus, TOY-D and TOY-B gene sequences were used in a blast pieces of the entire genome. The gene cluster was then found, which contains TUB-C, which has a 64% identity to TOY-D, and TUB-A, which has a 65% identity to TOY-B. To confirm the tubercidin production capability of the proposed gene cluster, a heterologous host was transformed using a cosmate with tubercidin gene cluster. The resulting metabolite was synthesized in the following LC-MS analysis. The first result is the authentic standard of tubercidin, so that we will know what tubercidin looks like. The second is the metabolite of the wild type S. tubercidicus. The third is the metabolite of the mutant S. collicolor that we are investigating, and the fourth is just a negative control. As you can see, the spike in the value of the third is very similar to the first and second. So we can be sure that the metabolites of the transformed S. collicolor really is tubercidin. So tuberculous color, which previously wasn't able to produce tubercidin, became able to after being transformed with the cosmate that contains the TBN gene cluster. With these results, we can be confident that it is very likely that this gene cluster is indeed responsible for the tubercidin biosynthesis. As for the in silico analysis, TUB-CBA were identified to be responsible for the synthesis of CDG. TUB-D is responsible for the synthesis of IMP, and several of these enzymes have been linked to toyocamycin synthesis. Others have unknown roles or are related to diazepurin synthesis.
So the gene TUB-E encodes a purine PRPP transferase. In vitro testing showed that there was no product generated after treatment with diazoguanine and PRPP. However, re-examination with CDG causes the presence of a new peak at the 24.7-minute mark, as indicated by HBLC results. LCMS results also showed a presence of a characteristic M+H ion peak at the 47.0591 mark. And MS spectra revealed the presence of fragment ions as shown in diagrams DNA, and the obtained hydrogen-1, carbon-13, and 2D NO spectra were consistent with those compound number 1, indicating TUB-E catalyzes PRPP transfer into CDG to form one of the intermediates in TBN biosynthesis. And in silico analysis indicates that the ambidihydrogenase GMP reductase domain is present with TUB-D, meaning the enzyme have a similar role in TBN biosynthesis. LCMS results indicates the presence of target IMP ion at the 349.0540 mark in the TUB-D reaction. MS spectra also reveals the presence of fragment ions as indicated in diagrams B and C in this slide. And the LC- results indicates that only net pH works as the cofactor for the tube D reaction. And tube G contains a highly conserved 2 residue Nudix motif, meaning it is predicted to be a Nudix hydrolyse, catalyzing the final tailoring step in TBN biosynthesis. HPLC analysis indicates the presence of expected adenosine peak in the tube G reaction, but absent negative control. LC-MS analysis shows the presence of target MSH ion at the 260.1034 mark, and the fragmentation pattern is consistent with those of adenosine standard. And furthermore, the cobalt ions were shown to confirm maximal activity of the tube G enzyme.
Here is the final proposed pathway for the biosynthesis of tubercidin, which shares the same initial enzymatic cascade found in the pathways of other antibiotics containing diazepurins, with the conversion of GTP to CDG. CDG is then converted to compounds 1, 2, 3 by the enzymes TUB-E, TUB-F, and then TUB-D. Compound 3 would then be converted to compounds 4 and 5 by the enzymes found in the main purine metabolic pathway. Compound 5 would then be converted to tubercidin by TUB-G. Tubercidin would then be phosphorylated by TUB-H, which would prevent it from attacking the cell when it's transported outside later on by TUB-I. So in conclusion, the tubercidin pathway is finally discovered with the roles of enzymes and cofactors found, and in the future, this pathway may serve as a template to synthesize new antibiotics. It's recommended that the other antibiotic pathways also be discovered, particularly those that contain diazepurins, with the tubercidin pathway as a reference. That is all from us. Thank you.